Hi, this is the second part of the applied genetics in the laboratory animal science, namely the genetic quality assurance or genetic standardization. As you can see in the study plan, we are over in the part 1. We discussed and explained the cases of genetic uniformity. And now we continue with the cases of genetic variation, namely the random bred colonies and outbred stocks and mosaic populations, supplemented with the genetic quality control, the representation and calculation of genetic relationship between inbred strains, and the questions of the biobanking. It may be preferable to have a certain degree of genetic heterogeneity within a group of animals, for example, the base population in selection experiment, or a background population for deleterious mutants like nude gene in mice and rats. In this case, the outbred colonies, like human populations, are heterozygous. They are therefore frequently used in toxicology and pharmacology research. But it is difficult to maintain a given level of variation within an animal colony than to maintain its uniformity. We'll see previously. Unless the number of breeding colony is infinite, inbreeding is inevitable, causing a loss of alleles in subsequent generations. So we need appropriate techniques. The first approach for the genetic variation is the random bred colonies and outbred stocks. The increase of the inbreeding coefficient, the delta F, when mating occur at random per generation is according to the previously shown equation, delta F equal to 1 per 8 female plus 1 per 8 males number, or if the number of males and females is the same, 1 per 2n, n being the population size, and it is supposed that the population is, according to Hardy-Weinberg rules, an ideal population. The goal is, in this case, to decrease or prevent the increase of the inbreeding coefficient. So the equation is the same, but the goal is the opposite to the inbred colony, inbred strains production. Another danger of small animal size is the bottleneck effect. If the delta F of a random bred colony increases, the gene pool will also decrease. This process is accelerated if the breeding population reduced in size. It is the case in golden hamster population, which came from one family. Middleston Colonel took with him from Syria, or in case of a change of the microbiological status from conventional to SPF by caesarean section. But in case of the cheetah, this is an excellent model to see the bottleneck effect in the nature. Scientists have witnessed population bottlenecks in American bison, greater prairie chickens, northern elephant seals, the mentioned golden hamsters and cheetahs. The New Zealand black robins experienced a bottleneck of 5 individuals, all descendants of a single female. Geneticians have found evidence for past bottlenecks in pandas, golden snub-nosed monkeys, and for the human population too during the long history. Here you can see a picture from the National Geographic, the genetic bottleneck, a threat to cheetah survival. Cheetah have suffered multiple genetic bottlenecks in the last 100,000 years. In today's rapidly changing climate, it's unclear if or when another bottleneck event will occur for those beloved cats and how they will adapt. You can see at the bottom the symbols of the genes, different colors, and from each bottleneck event, the number of color reduced. Solution to prevent the reduction of the gene pool are as follows. If offspring of as many parents as possible are being used for breeding, but this is only in the theory and in the dreams. If the matings are not random, breeding partners may be only distantly related. So this is a paradoxon that this random population cannot be crossed randomly. Rotation schemes may reduce the delta F by 50%, rotation changing the generation, and the best is the MAI, the maximum avoidance of inbreeding systems. The gene pool gradually changed over time, and the reproducibility of the results of successive experiments will be limited. These are the backdraws of these stocks, that the gene pool gradually changed over a time, and the reproducibility of the result of successive experiment is limited. So we will find a solution with the mosaic population, see later. But returning to the MAI, the maximum avoidance of inbreeding, with the supplemented with gold standard program in quality control, you can see that each generation is a rotation and rotation. Outbred stock criteria. What are the criteria of outbred stock in the laboratory animal science? The colony is randomly bred, maintained as a closed population for at least 4 generations. The number of breeding animals is sufficient to guarantee that, and this is the most important, the delta F, the increase of the inbreeding coefficient less than 1% from generation to generation. And finally, no artificial directional selection for any characteristics, with the exception of breeding performance, fertility. Nomenclature for an outbred stock: breeder or holder name, colon, stock symbol of 1 to 4 capital letters, CRL, colon, CRL stand for Charles River, CD, BR, barrier reared, Sprague Dowley outbred rat, or CRL, colon, NMRI, BR mouse, SSC, colon, NMRI, SSC, Statens Serum Institute, Copenhagen. Important outbred stocks today are the ICR, this is a CD1, the CFW, and the NMRI. All the three derive from the original Swiss mice in the USA. And the SF1 mice, this is from a non-Swiss mouse. And SD Sprague Dowley, Wistar, and long evens rats are the most important outbred stocks today. During long-term experiments, in particular if consistent genetic variation is essential, the use of an outbred stock is inadequate due to the gradual change of the gene pool. Inbred strains show different pharmacological, toxicological answer and sensitivity. This picture, this table shows only this is an illustration for the previously described different pharmacological reaction of inbred mouse strains. You can see that in the different strain are very different in insulin sensitivity, in histamine sensitivity, and in during the pregnancy the cortisol application caused cleft palate incidence from 19 to 100%. This is a teratological changes in the fetus. So we can conclude that we need some solution because the toxicology and pharmacology need a wide variability of a wide range of alleles, but a repeatable population, a uniform population. So this is the so-called Hungarian saying that from wooden how to make a iron seal, but this is possible, and this is in the laboratory animal science, the mosaic population, the inbred strains and their F1 hybrids. The aim is clear for everybody. The Hungarian Esther Csolnoki developed this system. The genetic variation can be standardized by using reciprocal crosses of a number of inbred strains, and this will assure in pharmacological toxicological tests on a widely variable gene pool. Same genetic variation can be generated at a later stage, anytime, if you conserve the founder inbred strains. The realization is through reciprocal crosses like the national soccer competition or championship. The number of genotypes is n plus n on the second minus n per 2. For example, if you have 4 inbred strains, the number of genotypes is 10. This calculation does not consider the so-called maternal effect, that is, we calculate as if AB is equal to BAF1 generation. Here you see the mosaic population, so the national competition, female and male from A, B, C, D genotype. A is not crossed with A, but crossed with all the other, and B is not crossed with B, but crossed all the other, and etc., etc. And if you take the same amount of individual from each genotype, this is the mosaic population. Unfortunately, changes in the genetic constitution of inbred strains produced by genetic contamination. This is, in other words, a miscegenation. It means the accidental mating of individuals from one inbred strain with animals of another origin. But we can have spontaneous mutations. We can have genetic drift and undesirable passenger mutations. The genetic drift have been mentioned. This means, to repeat, undetected fractions of alleles may become progressively fixed in a homogeneous state, replacing the original alleles. This process called genetic drift. And what is the undesirable passenger mutation? These are mutations that are hidden in the genomes of substrain of genetically altered lines and can affect the outcome of an experiment. So this is the reason that we need genetic quality control. Historically, it is possible to make by skin grafting, and the acceptance or rejection of grafts depends on the major histocompatibility complex of the donor and recipient. The genetic quality control can be made by biochemical markers too, and using electrophoretic techniques. These can be used to separate variants, for example, of isoenzymes. Each isoenzymes exhibit a different mobility when subjected to electrophoresis. After activity-specific staining in the homozygous animals like AABB, only one form of the protein capital A or B can be detected. In the heterozygous animals, AB, both zone A and B protein are present. So here you see the mouse isocitrate dehydrogenase gene and isoenzyme electrophoretic picture. The genetic quality control is possible also using DNA markers. The first possibility is that the polymorphism at the DNA level can be detected by restriction fragment length polymorphism plus PCR and gene electrophoresis. And it is possible to use the simple sequence length polymorphism, which technique came from the existence of microsatellites. Microsatellites are fragments of DNA consisting of a random repeat of 2 to 4 nucleotides at any specific locus. The number of repeats may vary between inbred strains. Some word about the microsatellites or short tandem repeats like guanine-adenine are repetitive DNA elements scattered throughout the genome, mostly in non-coding regions between genes or within genes in introns. Many are important in the variations in the number of repetition which can generate different alleles. So the number of repetition is different and it will generate different alleles. They have high mutation rates, which therefore they are highly polymorphic, and they are appropriate molecular markers. Therefore, they can be used for DNA fingerprinting and identification purposes. Many microsatellites are located mostly in non-coding DNA and are biologically silent. Others are located in regulatory or even coding DNA. Microsatellites mutations in such cases can lead to phenotypic changes and diseases. A genome-wide study estimates that microsatellites variation contributes 10–15% of heritable gene expression variation in human. Here you see as an illustration, right a older picture and left a new one. The different position of the bands indicate a difference in length of the microsatellite in an inbred strains. So the SSLP is made visible through the combination of PCR and gene electrophoresis. This is a good example of genetic contamination detected by SSLP PCR. The picture shows a 4% agarose gene with the characteristic bands obtained after PCR amplification using genomic DNA from 4 mice supposedly belonging to the bulb C strain, first 4 lanes, plus a standard DNA control for bulb C, last lane. In this example, only 5 SSLP loci are shown, located in chromosome 1 to 5, Note the presence of heterozygosity, second and third bands, and homozygosity for band band 4 and 3, third band. So this is absolutely not a good result. That this is a clear case of loss of authenticity due to genetic contamination. The PCR products are compared with a 100 BP DNA ladder. So this is important in today. And finally, you can see how to represent the genetic relationship between inbred strain, how to calculate and how to represent. The genetic distance is estimated from the degree of alleles, concordancy and discrepancy from a marker genes or single nucleotide polymorphism from a lot. Neighbor joining distance tree of the mouse strains phylogenetic distances. This is Beg albino 129 and the DBA related strains in the number 1 group, number 2 group Swiss mice and Asian strains, and group 3 wide derived strains. And last but not least, the biobanking questions. If you have to preserve the genetic material XC2, X the original animal, it can be made in vivo in another animal population and in vitro. What to put in the biobank? The whole genome, embryos, or individual genes. How to do it? By mostly cryptopreservation, maintenance under liquid nitrogen in 196 degrees Celsius. It works well for gametes, embryos, and somatic cells. As a very good example is the artificial insemination history in case of the dairy cattle. But you can preserve individual genes in tissue culture introduced after introducing by a virus vector. The take-home message is that we may have two directions in the genetic standardization, namely the genetic uniformity using inbred strains or F1 hybrids of inbred strains. We can have genetic variation using random bred colonies and outbred stocks, and we can produce mosaic populations. Here you see the most important sources, also proposed for further reading, some important website, and at the end I simply thanks for your attention in this very, very important but not easy to assimilate topic.